So at this point, we've talked about chromosome structure. We've talked about inheritance of chromosomes and genes. And so today, what I want to do is actually look at how the information gets from the chromosome, gets from being a DNA nucleotide sequence to the characteristic, whatever that characteristic happens to be, dark hair, dark eyes, whatever it is. So we need to look at that process. How does the information get from chromosome to the real world and the reality of the organism? So we've talked about DNA structure already. You've seen this diagram before. The DNA double helix forms the chromosomes. Chromosomes are composed of a double helix of DNA formed of repeating nucleotides. We've talked about directionality, nucleotide sequences being significant. And so continuing that discussion then, when we talk about DNA, it's one of two types of nucleic acid, so we've talked about that structure in the past, and today I want to add in another nucleic acid, which is RNA, ribonucleic acid. So as we look at the sequence of information, then we're looking at how information gets from a DNA nucleotide sequence, that translates to an amino acid sequence, which determines protein shape, protein function, and the protein function then determines the characteristics of an organism. So as we look at this flow of information, this is a diagram I'll come back to a few times, but it begins with the DNA nucleotide sequence, and we're going to talk about a process in which DNA is copied into a similar material called RNA. So different types of RNAs will be generated from DNA. And this will be done in a process known as transcription, and that takes place within the nucleus of a eukaryotic organism, within the nucleoplasm. And then the RNAs then can diffuse through the nuclear envelope into the cytoplasm of the cell. And there, a protein will be made using the RNAs, and also amino acids will have to be introduced. But this is a process known as translation, also called protein synthesis. So transcription occurs in the nucleus, translation or protein synthesis occurs within the cytoplasm. And then that protein then can become, the protein then can become part of the structure of the organism. The proteins may do the work of the cell, be the enzymes that carry out and the work of the cell or chemical reactions within the cell. And also some proteins are regulatory. And so these proteins can feed back on the DNA and the RNAs as well to control expression of genes, determine which genes will function and which genes will not be functioning. And the RNAs themselves also affect regulation of genes as well. So RNAs can kind of affect regulation of other RNAs, and RNAs can also feed back on the DNA and determine whether a gene is going to be expressed or not. But we'll get back to that later. So again, looking at RNA then, the RNA structure of RNA is similar to DNA. One of the differences involves the sugar. So here the sugar is going to be a ribose sugar instead of a deoxyribose sugar. They really just differ at this one carbon, so OH here as opposed to a hydrogen here. And just as we numbered the carbons in DNA, we do the same kind of thing with RNA because directionality is important. So we'll have our carbons 1, 2, 3, 4, and 5. And again, they're composed of nucleotides, and so the nucleotides then can be strung together, the sugar phosphate backbone with nitrogenous bases coming off the side. And so again, it's going to be important to know, which is the 3' end, which is the 5' end, because that can determine the order in which we read the bases, and then that in turn will determine what the amino acid sequence is of a protein. But we'll look at examples of that in a few minutes. And so then another difference in RNA and DNA involves what types of nitrogenous bases are used. So you may recall in DNA structure, the 4 bases are adenine, guanine, cytosine, and thymine. And in RNA, it's going to be adenine, guanine, cytosine, and uracil. Uracil takes the place of thymine. So any place that you would normally use thymine in DNA, uracil is used in the place of RNA. So when we look at complements that can form then, in DNA always forms an adenine-thymine complement, and guanine-cytosine will be complementary to one another and form hydrogen bonds. But in RNA, guanine-cytosine will still form the complement, but it will be adenine-uracil that form complements. And RNA can form complements with other RNAs, or sometimes portion of the same RNA, or RNA can form complements with DNA. So we sometimes get DNA-RNA hybrids. And again, we'll talk about that in just a second too. So again, as we look at differences between DNA and RNA, so DNA uses deoxyribose sugar, RNA ribose sugar. The nucleotides in DNA, adenine, guanine, cytosine, and thymine, whereas in RNA it's adenine, guanine, cytosine, and uracil. Again, leading to the pairings that we just discussed. And then also then when you look at a strand of nucleotide, even if I don't show you a lot of detail, you can determine whether it's a DNA or an RNA strand. So looking at this strand, this is a DNA strand because we can see that there are adenines, guanines, cytosines, and thymines. So when you see thymine, you know this is DNA. And we could form an RNA complement with this. And we'll see how this happens in a second. And what you'll notice then is that any place where thymine normally would have been found, uracil will go there. So again, we would know that this is an RNA strand because we see that there are uracils in the place of thymine. And again, hydrogen bonds can temporarily hold these together. Now, another thing to know about RNA, especially when RNA forms a hybrid with a DNA like this, is that RNA does not form as strong of a complement with DNA as another strand of DNA would. So this usually doesn't hold very well. So typically these strands of RNA will peel away from the DNA strand. So these are kind of temporary adhesions that we get between these two. And then DNA also tends to be very long. In humans, for example, if you put our 46 chromosomes end to end, they would be more than 6 feet long. They would be basically almost as long as I am. Whereas RNA strands tend to be very short. Also, DNA is confined to the nucleus because of the length of the chromosomes. The chromosomes are so long they really can't diffuse out into the cytoplasm, but DNA strands being short can move into the cytoplasm. And then also structurally, DNA normally forms a double helix, whereas RNA is normally single-stranded and can fold back on itself and form some very complex three-dimensional structures. So again, as we look at this information in, basically the information is in the DNA. Complements are made of RNA because those can get to the cytoplasm, and that's where proteins will be generated that, again, make up the structure of the cell, do the work of the cell or organism, or regulate and determine which genes function and which genes don't. So looking at RNA, there are many types of RNA. We're going to look at just a few types of RNA, those that are directly related to how proteins are made. So the first of these is what's known as messenger RNA, or mRNA. Messenger RNA is composed of codons, and a codon is a sequence of RNA composed of 3 successive nucleotides. So three nucleotides in succession form a codon. One codon codes for one amino acid. There are 64 different codons because this is what we call a triplet code. There are 4 possible nucleotides, adenine, guanine, cytosine, and uracil. And you're going to take those 3 at a time in a codon. And so if you take 4 to the 3rd power, there are 64 different combinations, meaning there are 64 different possible codons that you could have in mRNA. Also, I think what's important to know about these codons is that it's a universal code. So a particular codon codes for a particular amino acid. And this code for the mRNA codons is universal. And so this is a chart we'll be using in a second, but part of what I want you to understand is it's showing us the 64 different codons here. And so what I mean by universal code is that when we look at mRNA, if I look at 5' CCU, that's going to code for proline. That's going to code for proline in a human. That's going to code for proline in a bison, elk, aspen, willow, the bacteria. It doesn't matter what organism we talk about in the Yellowstone ecosystem, they're all going to use exactly the same code. So if we know the nucleotide sequence of an mRNA, looking 3 bases at a time, 3 nucleotides at a time, we can determine the amino acid sequence of a protein. And we'll look at examples of that in a second. Now, another type of RNA is transfer RNA. Transfer RNA positions amino acids for bonding and forming a protein. So they possess an anticodon that's complementary to a codon. We'll see how that works in a second. And as I mentioned, they position amino acids for bonding and protein synthesis. And then ribosomal RNA is a component of ribosomes. And ribosomes coordinate the activities of mRNA and tRNAs in protein synthesis. So again, where we're headed, DNA codes for RNAs in a process known as transcription. RNAs move into the cytoplasm, and in a process of translation, a protein will be made. And that protein then will either be part of the structure of the organism, do the work of the organism, or feed back and regulate expression of genes. So let's look at an animation first, which shows us these processes of transcription and translation. So looking at transcription, we're moving into the nucleus of the cell here. And then once inside, of course, we're looking at a chromosome, and this is a really important molecule here. This is RNA polymerase. And so the RNA polymerase makes complements of RNA. So it reads the DNA strand. It separates the DNA double helix. It reads the DNA strand and then binds complementary nucleotides of RNA. So whereas DNA polymerase was something that was used to make new chromosomes, RNA polymerase is going to read existing DNA strands and then build an RNA complement from that strand. So again, what the polymerase will do is uncoil the double helix, read nucleotides of DNA, and put in and build a complementary strand of RNA. And actually, I'd like to go back and show you that transcription one more time. So looking at transcription then, we're going to enter the nucleus. We're looking at the nucleus. Here comes RNA polymerase. RNA polymerase is going to bind to the DNA strand, and it starts to build an RNA strand from that. And this is going to be the messenger RNA in our particular case. So it moves along, reads DNA, and then inserts complements of RNA. So this then leads us to the next part of the process, which is translation. So as you can see, that RNA strand was beginning to peel off, and it will peel off from the DNA double helix. It's processed. We're not going to worry too much about this processing that goes on. And then that RNA strand will diffuse into the cytoplasm of the cell outside the nucleus, and this is where translation occurs. The protein synthesis takes place. And so what's going to happen here is that we're looking at tRNAs, and the tRNAs possess an amino acid. And so here's one of the subunits of a ribosome attaching. Another subunit of the ribosome attaches. Here's a tRNA holding an amino acid. Another tRNA will bind, and that positions the amino acids for bonding together. So they bond together, and now this is 2 amino acids, the shortest possible protein. The ribosome will shift one codon. That allows another tRNA to come in, positioning another amino acid, allows bonding together. And so the amino acid chain begins to grow. So what's important to understand, though, is that what's happening is that the mRNA has a sequence of nucleotides. 3 nucleotides in succession is a codon. The tRNA has a complementary sequence of bases. That's an anticodon. And so depending what the anticodon is of a tRNA, that will hold a particular amino acid. So a particular codon, a particular sequence of nucleotides, corresponds to a certain anticodon on tRNA, which will correspond to the positioning of a particular amino acid. So the sequence of codons, sequence of RNA nucleotides, corresponds to an amino acid sequence which forms a protein. So again, we're looking at a codon, the complementary anticodon. And the amino acid and the tRNAs can be reused over again. They'll be charged with different amino acids. The ribosomes can be reused again, and the mRNAs can be reused again. And again, as you can see here, what's happening then is as the mRNA sequence is read, the amino acid sequence grows. This eventually peels off and forms the protein. All right, so let's look at this in a less tech example, I guess, but let's go ahead and look at how we can take a DNA double helix, which I've unwound here, from that, transcribe that into an mRNA strand, and from that then determine an amino acid sequence. So again, what RNA polymerase would do is separate the DNA strands, and it would read one strand or the other. And so we're going to be reading this strand here. So we're just going to ignore this other strand. We're going to pretend it doesn't exist. And so again, RNA polymerase would build a complement of RNA. And this will be an mRNA. So we're looking at an mRNA strand here composed of a complementary sequence to a DNA strand. So here's our mRNA. And so we're going to look at 3 bases in succession. And what's important to know is that the code here we read 5 to 3. So we're interested in the chemical direction, not the physical direction. So we're going to start at the 5' end of the sequence and read to the 3' end of the sequence. And so if you look at the first codon is 5' AUG. And so a ribosome would bind there, tRNAs would be positioned holding an amino acid. But the point is that we can go to our code and see what amino acid should be in that position. So the way that we read this is that the first codon in the sequence is here, second codon in the sequence is along the top, and the third codon in the sequence is along the side. So the first sequence, or excuse me, the first codon in eukaryotes is always 5' AUG. So first codon A, second codon U, third is G, and so that's going to be the amino acid methionine. So methionine then would be positioned first. Then the ribosome will shift one codon. It will match up the appropriate tRNA, and that codon is 5' GUA. So if we go to our chart, GUA is going to correspond to the amino acid valine. The ribosome will shift one codon, and we have CGG, and that's going to translate to the amino acid arginine. Then the codon 5' UAC, UAC, that's going to correspond to the amino acid tyrosine, UCC, translates to serine. And then in our particular sequence here, UGA is one of 3 codons which are known as stop codons. So 61 of the codons code for amino acids. 3 of them are what we call stop codons. That will result in what's known as a releasing protein sitting in that position, and that will cause the ribosome to release our protein. Because again, what we have now is by bonding amino acids together, we have a protein. So what we have then is a DNA sequence, which translates to an RNA sequence of an mRNA composed of codons. And then by reading that nucleotide sequence 3 at a time, or 3 in succession, I guess I should say, that will determine an amino acid sequence. And that becomes the new protein. So I also just want to emphasize again that you're always reading 5 to 3 in these situations. That's kind of the standard. So it doesn't really matter what the physical orientation of the strand is, whether it's up, down, left, or right. You just want to be reading 5 to 3. And then from that, you can use your chart to determine what the amino acid sequence is. All right, so then mutation is important. So mutation is a change in the sequence of DNA. And I should say it's a random change in the DNA nucleotide sequence. And when we look at mutations, some mutations are what we describe as being heritable. A heritable mutation gets passed to the next generation. There are also somatic mutations. Somatic mutations are those that we say occur within the body but do not go to the next generation. So again, you might have a mutation in a liver cell that leads to cancer. That might be really bad for you personally, but at least it doesn't go into your gametes and go to the next generation. So the heritable mutations are significant when we talk about evolution. And it's mutation then that generates new traits in nature. So again, we're looking at, if we look at the sequence then, if you have a change in the sequence of DNA, that potentially changes the sequence of nucleotides in your RNA, particularly mRNA, which will change the amino acid sequence of your protein, which could affect the function of the protein. So causes of mutations. Direct damage to DNA strand by radiation or chemicals, what we call polymerase misreads. Also repair mistakes lead to mutations or changes, spontaneous changes in the sequence of DNA. So what I mean by DNA polymerase misreads is that when chromosomes are replicated in mitosis and meiosis, and we've talked a little bit about this already, you may recall that DNA polymerase reads the existing strand and matches up the complementary base. Well, normally it's very faithful in doing that. We have a very good DNA polymerase. It rarely makes mistakes. But occasionally it does. So it is possible that DNA polymerase, as it's moving along a strand and building a complementary strand, will make an abnormal chromosome. So these misreads then would just be, well, what if it just made a mistake? What if it accidentally read or couldn't figure out what a particular nucleotide was and didn't know what to put opposite that? Then you might end up with mistakes in your sequence of nucleotides, which could affect protein shape, protein function. So we'd refer to those as misreads. Now, the consequences of misreads are that you could have a nucleotide substitution, having one nucleotide substituted for another. You may actually end up with nucleotide additions or nucleotide deletions, and all of those have consequences. So let's look at what we call point mutations. So one type of a point mutation affects a single nucleotide. So one type of a point mutation is a missense mutation, another is a silent mutation, frame shift, and nonsense mutation. So let's quickly look at examples of each of these. In a missense mutation, we're going to target a particular nucleotide here. What happens is that we'll have some change in that nucleotide sequence. And if we look at the original, what it should be, C should transcribe to G, and that would lead to a valine in this position in our second codon here. What if something happened to this cytosine so that it was replaced with a guanine? Well, that would downstream would affect a cytosine being built in and the mRNA. And the question is, would that still be a valine in this position if that happened? And the answer is, in this case, no. Actually, that would lead to a leucine being inserted into that position. So we've changed the amino acid sequence. That could change protein shape. That could change protein function. There's no guarantee that it would, but it could. Let's go back to our original strand and look at what's known as a silent mutation. Here we're going to target this thymine. What if this led to some change, maybe a misread or whatever, so that that thymine then is changed to a cytosine? Again, that would have a downstream effect. That would change this to a guanine. And so the question again is, would that change the amino acid sequence in this case? And in a silent mutation, the answer is actually no, it would not change the sequence. So we would still end up with a valine there. So a silent mutation is where you actually do have a change in the nucleotide sequence of the DNA, but it does not lead to an amino acid change. And the reason for that is that when you look at the codons, there's often more than one codon coding for the same amino acid. Okay, a frame shift mutation occurs when there's either an addition or a deletion of a nucleotide. So again, if we look at this cytosine here, what if something happened so that that was literally just knocked out of the strand? So initially we have our codons, so we have AUG, GUA, and so on. But again, what if something happened so that one of those nucleotides was eliminated and then the strands spliced together? That would change all of our codons here. So we would be shifting everything here to the left so that our codon is changed here, this codon is changed, this codon is changed, this codon changed. Anyway, that's going to have huge downstream effects. So that's not going to affect just a single amino acid, that's going to affect a number of amino acids. So we would actually have a complete change in the amino acid sequence. So this really isn't just a small change in a protein, this is a gross change in a protein. And again, would it have a function in nature? I don't really know. Nature would decide the value of this new protein, and nature always decides the value of mutations as well. And then a nonsense mutation is one in which the change causes the insertion of a stop codon. So if it's a mutation that leads to a stop codon being produced, that then can change protein production, and instead of the whole protein being produced, only a fragment of the protein will be produced. And again, that would be a nonsense mutation. So again, when we look at what leads to actual damage to the DNA strand, some of it is radiation. Radiation damage can literally knock out nucleotides. We have enzymes that will come in and repair those, but they don't necessarily repair faithfully. So you can end up with mistakes being made there. UV radiation also particularly affects thymines, so that thymines will begin to form covalent bonds with one another. This kind of puts a bulge into the strand of DNA. And again, this can lead to misread problems when polymerase comes along. Polymerase comes along, it's not sure what to put opposite that, or also when that, what we call this is a thymine dimer, when this dimer is repaired, there may be mistakes that are made there. So again, those repair enzymes may not repair properly. Now, what can also lead to these misread kind of problems are chemicals. And so as we look at chemicals, one type of a chemical mutagen would be what we call intercalating agents. Intercalating agents can wedge in between nucleotides so that when DNA polymerase comes along to read this, it's not sure what the nucleotides are, and it will just randomly insert complements in there. So that can lead to problems. Also what we call base modifiers. Base modifiers bind to nucleotides, and again, as polymerase goes through here, it reads this. It can't really tell what should go opposite that, and so we end up with mistakes being made in our complementary strand. And then also there are what we call base analogs. In the construction of nucleotides, sometimes there are chemicals that look like the normal bases, which aren't, and so they actually are included in the structure of a nucleotide. And again, as DNA polymerase is moving through, it's not sure what that is, and it makes mistakes when it builds in the complements. We can also have chromosomal level abnormalities. And these occur primarily in meiosis when the gametes are being made, but there's some other types as well. One of these is what's known as jumping genes. There are genes that code for enzymes that will cut those genes out of one chromosome, and then those can be spliced into another chromosome. So these transposons or jumping genes then can randomly be kind of jump from one part of the genome to another, and they can cause disruptions in our genome when they do that. Again, during meiosis, gamete formation, there's a point in that process in which the chromosomes must all be untangled and separated from one another, and a lot of times breakages of chromosomes occur. So we can have what we call translocations, where a breakage of one chromosome occurs and it's spliced onto another chromosome. It may be spliced onto the end of a chromosome, or it may be spliced into the middle of a chromosome. This is the more problematic situation, because if you had some gene here that was coding for a protein and you arbitrarily splice in something else, that's going to disrupt the protein that should have been made. We also get what we're called inversions. Again, this would also be occurring during meiosis, during gamete formation, where the chromosomes are being cut and spliced with one another. And sometimes cuts occur. The order of a chromosome may get flipped upside down, basically, or a portion of it, and then spliced back in. So instead of a particular sequence of genes being generated, we actually invert that sequence. Now, this isn't really a classic mutation in that we don't really necessarily have a change in the sequence of DNA, but we're changing the order of genes, and that can affect how they're used and whether they function normally or not. Normally, it's just important to have the genes, but sometimes the position of the genes is important. And then also what we call non-disjunction events can occur. So in other words, whole chromosomes may not separate properly. So we're looking at what's called a karyotype. At certain stages of mitosis, you can observe the chromosomes. So here we're looking at a normal human karyotype where we actually have 23 pairs of chromosomes, homologous pairs of chromosomes. And again, sometimes a gamete may carry an extra chromosome, or it may be missing a chromosome. And you can see in this karyotype, there's actually 3 of number 21. So that would be a chromosomal abnormality. So again, these downstream effects then are that this leads to abnormal proteins, and eventually nature has to decide the value of these changes, and that's what we'll get to in our next module. See you next time.